Hi, my name is Grant Kramer, and I am a professor emeritus at the University of Nevada, Reno. Water. It is the key to life on this planet. As you can see here at Lake Tahoe, it has three different phases. The vapor phase, which forms the clouds behind me in the sky, the liquid phase, which makes up the lake, and the solid phase, which is in the form of snow and ice on the shores of this beautiful, beautiful location.
Lecture number 2. In this lecture, we will discuss the importance of water, diffusion, free energy, and the water potential. So why is water so important? Well, first of all, plants are made up of 70 to 90% water in general. Water is the main limiting factor for land plants. In addition, plants have evolved multiple mechanisms for acquiring and keeping their water. However, 99% of the water absorbed by a plant is lost to the atmosphere. We are going to find out why. In fact, water availability really leads to a loss of yields in crop plants. Here in this figure, you can see the corn yields going down in a field as the water availability decreases. And this is a general rule for most ecosystems, where we can see plotted here the productivity of an ecosystem versus the annual precipitation in that ecosystem. How is water transported or taken up? To understand this, we must first understand diffusion and the water potential. But before we discuss those principles, let's look at the characteristics of water molecules in general and see why it is such a special molecule and why it's so important to life here on this planet. So if we look at this diagram of a water molecule, we can see it's made up of one oxygen atom and two hydrogen atoms, and they form tetrahedral structure in their three-dimensional shape. Water molecules have two important properties that make it important in the plant. One is that water molecules are polar, and the second, which we'll discuss in the next slide, is that it forms hydrogen bonds. So what do we mean by polar? Because the oxygen atom is more electronegative than the hydrogen, it tends to attract the electrons of the covalent bond. This attraction results in a partial negative charge at the oxygen end of the molecule and a partial positive charge at each hydrogen, making this water molecule polar. These charges are counterbalancing so that the overall water molecule has no net charge. The opposite charges on the hydrogen and the oxygen molecule make them attractive to each other, but between other water molecules, and thus we get the formation of hydrogen bonds. These are electrostatic bonds, not covalent bonds, so that the oxygen atom of one water molecule is attracted to the hydrogen atom of another water molecule. These hydrogen bonds are responsible for many of the unusual physical properties of water. In addition to water hydrogen bonding between itself and another water molecule, it can also form hydrogen bonds with other electronegative atoms such as oxygen and nitrogen. And these occur in some of our organic molecules as well. In this next image, you can see that the formation of hydrogen bonds between water molecules allows some sort of structure to occur within a water molecule, so that you can see that there's almost a crystalline structure of these water molecules on the left-hand side. This, in fact, is what happens when water molecules become cooler and reduce their vibrational activity, and eventually, as the temperatures get cold enough, those water molecules form an ice crystal. However, they also form this random configuration because of the rapid kinetic energy, and they're forming and breaking hydrogen bonds all the time. Some additional water properties that occur as a result of these hydrogen bonds is that water forms a liquid at room temperature. The hydrogen bonds between those water molecules inhibit the gaseous evaporation of water molecule up into the atmosphere, forming a vapor rather than a liquid. Another important property of water is that it has a nearly constant volume. It's very difficult to compress water, and this becomes important later on when we talk about cell turgor in the plant, which allows a plant to be rigid and stiff rather than limp when it is dehydrated and it becomes wilty. A gaseous bubble in a liquid assumes a spherical shape. This is the result of the surface tension that acts on that liquid-gas interface. Water has an extremely high surface tension compared with other liquids at the same temperature. We can see that a sealed syringe with an air bubble inside of it can be used to create positive and negative pressures. So as we press on the plunger, we create pressure. As we pull on that plunger, we create tension. This will be important when we talk about water movement in the xylem in the stems of the plants, where there are the formation of gaseous bubbles, which can lead to cavitation, as we'll discuss, which is the breaking of the water column, or also lead to, in the night time, the refilling of that xylem vessel. This will become more clear later on in a future lecture. Some additional water properties are that they can form adhesive and cohesive bonds. Again, this is a result of hydrogen bonding, and this is important in long-distance transport up the xylem. On the right, we can see the application of hydrogen bonds of the water molecule to the xylem wall structure, which is made up of some various organic molecules of cellulose and hemicelluloses. And this attachment by hydrogen bonds to the surface of the xylem wall is known as adhesion. Cohesion or cohesive bonds are the bonds that form between water molecules. So forming hydrogen bonds between a water molecule. So when we say that a water molecule adheres to the wall of some structure, whether that's the xylem or a glass container, we're talking about adhesion. When we talk about water molecules binding to each other through hydrogen bonding, we call that cohesion. Another important property of water is that it forms a good solvent, which allows it to dissolve substances and minerals because it's able to neutralize the charges of those molecules and form cages around them. For example, here we can see a salt crystal of sodium chloride with water molecules aligning up to the sodium particle, which has got a plus charge, which results in the negative ends of the water molecule surrounding and caging that sodium ion and preventing it from binding back to a chloride ion, which would also be attracting water molecules, but in this case would be attracting the partial positive end of the water molecule on the hydrogen ends. And thus it forms cages around these molecules, or ions in this case, and prevents them from binding into each other and forming a crystal again. So this is a particularly good property of a solvent, to be able to dissolve those substances. And this is a result, again, of the polar nature of water molecules and the formation of hydrogen bonds. When a water molecule adheres to an object, it forms a sphere that has an angle to it, as seen in this picture here. On a substance that we would call hydrophilic, that is water-loving, it will create an angle that's less than 90 degrees. The angles that form on glass or on the primary cell walls of a plant cell, those contact angles will be close to zero. On a hydrophobic surface, those molecules will form a bead, and the hydrophobic surface has a contact angle that will be greater than 90 degrees. We will see that on the leaf surface, which has got a waxy surface to it. And that waxy surface is a hydrophobic surface or substrate, resulting in the beating up of water molecules on the surface of the plant. Cohesion and adhesion and the surface tension that forms from those create a phenomenon known as capillarity. When we look at this tube here, we can see that a meniscus forms in that tube. The meniscus is formed from the surface tension of the water, starting with the adhesion of the water molecules to the side of the capillary glass tube. The cohesive forces of water prevent the water molecules from being ripped apart as they bind to each other and thus pull all of the water molecules in the column upwards with this net force pushing upwards against gravity. The gravity pushes the center molecules down further than the ones on the edge because of the strong adhesive forces in the water molecule. And at equilibrium, this water column is formed higher than the level of the water in the beaker. The opposite occurs where a meniscus curves downwards in a liquid where it has a contact angle greater than 90 degrees, as in the case of mercury, with a contact angle that equals 140 degrees. Now we have gravity that is actually acting on a force on mercury, causing the mercury molecules to bend downwards or causing the meniscus to curve downwards. Okay, now let's talk about diffusion. Most of us know the word diffusion, but do we really understand the properties of diffusion and what controls it? Diffusion is defined as the movement of a molecule or an ion from one point to another by random kinetic energy. In this square here on the right, we have a number of blue molecules and a number of red molecules. And it's a great demonstration of what diffusion is like by the kinetic motion of molecules in a box. We can see that they are all moving around, and as they bounce into the walls, they bounce off, but as they bounce into each other, they also bounce off, just like a bunch of billiard balls. This is random kinetic energy of the molecules, moving molecules around, and eventually forming in an area, but never constant. It's always moving, and this is something to remember. Even at equilibrium, we will find molecules still moving around. It'll just be that at equilibrium, the net movement of these molecules will be zero. The kinetic theory tells us this motion is constant above an absolute temperature of zero. And it tells us that the velocity of these molecules is dependent upon the temperature and their molecular weight. So we can see that higher temperature equals to higher velocity, and a larger molecular weight would equal a lower velocity. So smaller molecules will move faster than larger molecules. Down in the bottom right-hand corner, we can see that diffusion leads in from one concentrated area to a more uniform area. And this is where we're reaching equilibrium of red molecules in this container of water or water molecules. So again, another image of diffusion where we can see a dye that's been dropped into a Erlenmeyer flask full of water, and we can just imagine the process of this diffusion of these molecules moving out throughout the water to become a uniform pink color. And that's demonstrated here in this image on the right, where we can see at the top beaker, the initial beaker, we can see that the beaker has red molecules on the left side and blue molecules on the right side. And we can see below it a graph of the concentration profiles of these molecules, with them both being at their highest, most concentrated amounts. As these molecules bounce around through random kinetic energy, eventually they begin to lower in their concentrations in the middle of the beaker where they are interacting with each other. And the exterior molecules, because they're just bouncing back and forth into each other, they're not yet mixing with the red and the blue, remain high in concentration. But with time, as that kinetic energy and that randomization continues, eventually we reach an equilibrium where we have equal concentrations of both the blue and the red molecules uniformly throughout that beaker. However, note that the ions are still moving around and there is no net diffusion. There are small movements of molecules in any one given area, but there's never any net change in the concentration of those molecules. So diffusion is occurring when a molecule is moving from high concentration to an area of lower concentration of that specific molecule, independent of the other molecules that are present there. And we call that diffusion. And the rate of diffusion is dependent upon that concentration gradient. That is, if there are concentrations that are very highly different, then there will be a rapid rate of diffusion. If they're very similar to each other, then that rate of diffusion will occur more slowly. And it's also dependent upon distance, with molecules moving very rapidly at short distances, but moving rather slowly over a long distance. This is reflected in this figure here, where we can see on the left-hand side a representation of the concentration gradient from low to high over a range or distance. So we can see the delta S, or that's the triangle, which is referencing the differences in concentration, over the delta X, which is representing, again, the triangle with an X that represents a change in distance. And then on the left-hand side, we see in this process this concentration gradient driving diffusion. So we have a very high concentration difference. It's very steep on the figure on A. When we go to the figure on B, after a period of time, we see that the overall concentration has declined, and the concentration gradients now have become much smaller, and thus the rate of diffusion starts to slow down. Plus, it's spread out over a longer distance than it was in figure A. We'll come to a mathematical equation to describe this in a moment.
Now, I know a number of you are going to say, wait a minute, equations? I hate equations. Well, I'm going to tell you that equations are beautiful. They're a way of bringing in information into a concise way so that you can understand what is going on. These equations are condensed bits of information. So bear with me as we go through some equations here in order to better understand diffusion, water transport. After all, science is about measuring things, and we need measurements and data to help us describe the events that are going around. And equations help to condense that into the variables that are most important.
Now, in order for us to understand diffusion, we also need to understand thermodynamics and the definition of a term called the water potential. And this slide here is in honor of Jack Dainty, who was a nuclear physicist in England, who was interested in biology, and he took approaches that had been learned in animal biology and applied them to plant science. And he has revolutionized our understanding of how water moves and why it moves in plants, which gives us a much better understanding of things and properties such as water absorption into the plant, also ion transport, as we'll see. And these factors are very important for plant physiology. So I just give a shout out here to Jack Dainty, who applied this theory well before I was studying plant physiology, but I have benefited, as have many other scientists, by his excellent reasoning and thinking. So to understand diffusion, we must know something about thermodynamics. It's the second law that's most important, but let's discuss what the first law is. The first law is energy is neither created or destroyed, it's merely transformed. The second law of thermodynamics is any system plus its surroundings tends spontaneously towards increasing disorder. And the key word here is spontaneously, as we will be talking in the future about passive and active transport. And this word spontaneously implies a passive process versus an active process where energy is required. So what is disorder? The measure of disorder we call entropy, and we use the letter or symbol S. If entropy is increasing, then we know that the process is spontaneous. That is, if disorder is increasing in the system that we are investigating, then we know that the process is spontaneous. If entropy or disorder decreases, then energy, either work or work plus heat or heat, has been put into the system to create this greater order of the system.
A good example of disorder is in the gardens around one's house, where over time things spontaneously go into disorder or entropy. And it requires work or energy to reorganize and put that garden back into shape. Entropy is like death. It is the natural decay or disintegration of our bodies or the universe. And it reaches that equilibrium state. Equilibrium is death in that sense of the system that we are looking at. And life is the opposite of that. It's the capturing of energy from others or the universe in ways that allows our single cells to be developed and organized into whole bodies.
Now, we say if entropy is at its maximum, that is, that disorder is at its maximum, then the system is at equilibrium. So when we think about the beakers with the different molecules in them, we had maximum disorder or maximum entropy at equilibrium, when those two molecules were uniformly mixed throughout the beaker. So how can we predict when a process is spontaneous? And why do we care?
We care because we want to understand the processes around us, and we can define active or passive transport by better understanding of this thermodynamic process. We'll find that water transport is always passive in plants, but that ion transport can be both active or passive. And this helps us to define what the mechanisms of ion transport or mineral uptake are in a plant.
So thermodynamics allows us to begin to understand this process. Is energy required, or can the process occur spontaneously? Will diffusion occur spontaneously? And the answer for that is only down its concentration gradient. If it moves against its concentration gradient, then it is not spontaneous. So when is a process spontaneous? Gibbs free energy, or G, is the measure of the maximum energy available to convert for work in a system. It is the currency we use for all energy exchange. It includes the internal energy of the substances, including its chemical bonds, its kinetic motion, and its electron energy. It also includes the potential energy of a molecule, and that potential energy can be converted to kinetic energy. So what do we mean by that? It's like the movement of a stone or water down a hill. That's a potential energy at the top of the hill being converted into kinetic energy as it moves down the hill. We can convert that movement of water down the hill into work, as in this water mill, or in the form of electricity, as in a generator or turbine. Likewise, the combustion of fuel can be converted into heat or work. This is a result of the internal bonds of those molecules and the release of that energy in those bonds. So it is the change in free energy that is actually important in a reaction, for a reaction to occur. And so it's this delta G, or the differences in Gibbs free energy, and this is approximate to the changes in the enthalpy, or the internal energy of those molecules or system, minus the temperature times the changes in the entropy of those molecules. So free energy decreases for spontaneous reactions. When the delta G is negative, the reaction is considered spontaneous and is a measure of the maximum work that can be done. When the delta G is positive, then the reaction is not spontaneous, and it is a measure of the minimum energy required for that reaction to go. But you can't measure free energy. However, we can measure the chemical potential, and the chemical potential is related to free energy. The chemical potential is the free energy per unit quantity of substance. In this case, the temperature is assumed to be constant to simplify this derivation. The chemical potential of a substance is the function of its concentration, its pressure, the electric field, and gravity when temperature is assumed to be constant.
This concept of the chemical potential is very, very important. I encourage you to memorize these 4 factors, as they are the total possible factors that can be used for a chemical potential of a substance. When we talk about water, we will use only 3 of them. When we talk about ion transport, we will add the electric field, which is not used in the water potential. We will also use it in photosynthesis when we talk about the electron transport chain in the chloroplast, and also for energy production in the mitochondria.
So the chemical potential of water is called psi sub W in this case, or the water potential. We use this Greek symbol psi to represent the chemical potential, and we use the subscript W to refer to the water potential.
In the next lecture, we will go more deeply into the water potential, and we will look at other factors that are embedded in the water potential equation that include the solute potential and the pressure potential. And these will also use the psi symbol, but they will have different subscripts, the subscript S or the subscript P.
So the water potential is a measure of the free energy per unit quantity of water. And in this case, the water potential is a function of 3 things only: its concentration, its pressure, and gravity.
Because water is neutral, the electric field has no effect on water, and therefore we can drop it from the chemical potential equation for water.
To further define the water potential, we must choose an arbitrary value of water potential to make a standard value. We define the water potential at atmospheric pressure and constant temperature. The water potential of pure water is equal to 0. This is defined at the atmospheric pressure at sea level. This means that all other water potentials are negative values unless pressure has been added to the plant in some way to raise the water potential value, or temperature for that matter as well. But in our considerations of water potential, we are assuming constant uniform temperature. So just remember that we will always see negative water potential values. And that means that the more negative they have, the less free energy they have. So water movement responds to differences in water potential. That is, the water potential gradients are important. So water moves from high water potential to low water potential, because high water potential, which is still a negative value, is going to have more free energy in it than that of a low water potential, which is more negative and has even less free energy. If the water potential gradient, the delta psi W, is equal to 0, then the system for this water is at equilibrium. I'll repeat that. We're at equilibrium when there is equal free energy between two different systems. That could be inside and or outside the cell, for example, or from one cell to another. So water potential gradients are the key to understanding water movements in and throughout the plant. Water potential gradients produce the driving force for diffusion of water. There are three major factors that influence the water potential gradients in plants. That is, there's concentration, or more precisely, activity of water, there's gravity, and there's pressure. So let's talk about water concentration for a moment and activity. So water moves from high concentration to low concentration. What do we mean by activity? Activity is what we could call the effective water concentration.
Not all water molecules have equal activity. Some molecules are adhered to the sides of a vessel or a tube, and this restricts their activity. The activity of some water molecules is affected by the solutes. When they form cages, then they are bound electrostatically to those ions, and this reduces their activity as well.
So we can measure something called the mole fraction of water, which gives us a way of measuring the activity of that water. And that's the number of moles of water in a given area or volume, and dividing it by the number of moles of the solute or solutes in that solution, and the number of moles of water. I'll give an example of this in a future slide to just show you exactly what I mean. In other words, as we add solutes, that's in the denominator, and that will increase or that will decrease its activity. The more solutes that are added in solution will decrease the activity of the water. What about temperature effects on water movement? Well, this is something that gets rather complex, and it's dependent in part on the state of the water. Cooler water vapor will move towards warm water vapor along its concentration gradient. Because it's vapor, the volume of water condenses in cooler air. That is, the spacing between water molecules is closer, therefore it's more concentrated. When water vapor is warmer, the water molecules expand in the gaseous form, and therefore it's less dense. So cooler water always moves towards warmer water because cooler water has more concentrated number of water molecules in the same volumetric space. In another situation that happens in real life as well is that we have warm water vapor or liquid, which is warmer than ice, which is cold and a solid.
Because ice is a solid, the water becomes very inactivated. So as warmer water is moving towards ice, it becomes inactivated. It moves from high activity to low activity. This is what will happen in real life when ice is forming near the plant or inside the plant, and water molecules will move towards that ice crystal. This we'll discuss more in the future when we get into environmental stresses and talk about freezing stress.
What about pressure? Yes, pressure affects water as well, and we'll see that water moves from high pressure to low pressure. We can use these properties to understand how we might measure the water potential. And so this is linked to vapor density and vapor pressure, which help us to define a water potential. If the water potential of a vapor is equal to the water potential of the liquid, then we say that the system is at equilibrium. And we could see such a situation here in the right-hand side in what could be a closed beaker, with water in the liquid form at the bottom of the beaker and we see blue molecules of water as a vapor above that surface. And every once in a while, a water molecule escapes the liquid and moves into the atmosphere, as represented by the up arrow. And every once in a while, a water molecule in the vapor phase moves down into the liquid and is bound by hydrogen bonds to the other water molecules and becomes part of the liquid. Now it's at equilibrium when the number of water molecules that are escaping the liquid equal the number of molecules going into the liquid and binding to the liquid. We call that equilibrium. And that's very important because that's when we want to measure a water potential number or get a measure of the water potential. We have to have a equilibrium state, which I'll explain in a moment. So the amount of vapor that is in equilibrium with liquid water is a function of its vapor density and pressure. And the amount is independent of any of the other gases in the vapor phase. So vapor pressure, there are three things that affect vapor pressure. That's temperature, pressure, and the solute concentration. This is why we learn something about the mole fraction. So we can calculate the vapor pressure of a solution using Raoult's law, which is known here as P, which is the vapor pressure. It's equal to X1, which is the mole fraction of the solvent, which we just learned the definition of, and the vapor pressure of the solution versus the vapor pressure of a pure solvent. So vapor pressure is a product of the mole fraction of the solvent times the vapor pressure of the pure solvent. So let's give an example of how we can calculate this and why it's important. So we know that 1,000 grams of water, which is approximately a liter of water, is equal to 55.508 moles. And we know that a mole of sucrose is equal to 342.3 grams of sucrose. If we were to calculate the mole fraction of a solution of 342.3 grams of sucrose with 1,000 grams of water, we would show this equation here, or the X of water, or the mole fraction of water, is equal to 55.508 divided by 55.08 plus 1. Remember, it's number of moles of solutes plus the number of moles of water. If we calculate that out, it comes out to be 0.9823, or 98.23%. Now, we also know that the equilibrium vapor pressure of pure water is also equal to 100% relative humidity. That's when we reach equilibrium of a water in a beaker, for example, that's closed off. So effectively, the mole fraction of water is equal to the relative humidity of the air at equilibrium. This is where we get to define one form of the water potential. So if we look at the equation below, we see that the psi sub W, or the water potential, is expressed in terms of pressure, known as megapascals. This is a scientific term. We could be using bars or atmospheric pressure, but we use megapascals today. And it's equal to RT divided by V with a bar over the top of it, which is known as the partial mole volume, times the natural log of the vapor pressure divided by the vapor pressure of pure water. So we can say that the water potential is equal to RT over the partial mole volume of water times the natural logarithm of the relative humidity inside a container, for example. And we know that RT, and I'm just giving you this, so you don't need to actually go and calculate it. We know that the RT divided by its partial mole volume is equal to 135 megapascals at 20 degrees centigrade. And it's possible in the future I may give you an example to calculate out. And if I did, it would be at this 20 degrees centigrade. So all you need to know is the 135 megapascals for R and T. So the practical outcome of all of this vapor pressure theory is the fact that we can place leaves inside an enclosed chamber that is sealed. And by measuring the relative humidity inside that chamber, after it's reached equilibrium, we can establish what the water potential is. And as a result, there's been devices that have been developed that are called psychrometers. And psychrometers are devices which originally, like a sling psychrometer, were used to measure wet and dry bulb temperatures to determine what the relative humidity of our atmosphere is, or its vapor pressure. However, with these psychrometers, as shown below, we see that the leaf or a stem is placed inside the chamber that's sealed, and we give it enough time to allow it to reach equilibrium so that the water vapor inside the chamber comes to equilibrium with the liquid water inside the stem or the leaf. And we can measure the water potential of that stem or that leaf. That is the amount of free energy of water that can be used to do work. Why is that important? Because it's going to be able to tell us, can the plant spontaneously absorb water from the ground or not? Or is it going to lose water from the plant and move and be transported back out into the ground? This is extremely important, and scientists need to know this. They need to know whether they should be irrigating their crops, or actually it should be the farmers that want to know this. The scientists just are providing a way here to help the agriculturalists understand how they need to manage their crops. So inside this chamber, if you're curious, is a thermocouple, and this thermocouple is measuring differences in temperature change that occur because if the water potential was higher in the stem, for example, than on the thermocouple itself, the water would move to the thermocouple and condense onto it, and as a result, there would be a temperature change, and the thermocouple detects that. And vice versa. If the water potential was lower than what's on the thermocouple, then water would evaporate off the thermocouple, again changing the temperature, cooling it, and that would be detected by a meter. On the right here, you can just see a couple of examples of real psychrometers, one that's a little larger than the other, the stem psychrometer having a larger chamber than a leaf psychrometer. So before we go much further, let's talk about a few useful water transport equations that'll be useful for us in understanding how water is moving in different parts of the plants at different times, because it's going to move by different mechanisms at different times. For example, if we look at Fick's first law, which is the first law, is the law of diffusion, we can define Fick's first law here as J, which stands for flux, or the movement of a molecule, in this case water. And it's equal to negative D, D is the diffusion constant, times the change in C over the change in X, which is the concentration gradient. The change in concentration over a change in distance. So as the concentration gradient goes higher, the flux is faster. The transport, the movement is faster. And it's dependent upon the diffusion constant. And I won't go into the definition of a diffusion constant, but in part it's dependent upon the size of the molecule. So if we looked at a common example here of water diffusion, we could see that if we made the calculation that water moving across from one side of a cell to another that is 50 micrometers wide, it would take only 2.5 seconds for that water molecule to get from one side of the cell to the other. But if we looked at a 1 meter long branch, that water molecule would take 24 years for it to diffuse all the way across that 1 meter branch from one end to the next. Now clearly the branch would be dead before the water got there. So it's clear that water may move easily by diffusion within a cell, but cannot explain why or how water moves from one end of a branch or one end of a tree to the other. Here are a couple of other flux equations I want to give you before we give an example of how water is taken up into a cell. So again, we use the letter J, and this is Poiseuille's equation, and this will become important when we talk about bulk or mass flow through a pipe. In this case, we will have pipes or tubes in the form of the xylem structure in the plant where water is moving as if through a water pipe. And therefore, this equation becomes very relevant for that water movement. And in the case of phloem, I should add, which is also the movement of water moving through pipes, but in this case, it also has sugar in them. It's the main transport pathway for sugar transport. We have largely this flux is dependent upon the pressure gradient. So we have a change in P or delta P over delta X as the pressure gradient. And as the pressure gradient increases, we get an increase in flow rate. And this makes sense. You all know or have seen a fire hose and how it moves so much water because of the high pressure pushing that water through the tube. The other terms here are terms that are referring to the viscosity of the liquid, in this case, water that's being moved, the radius of the pipe. So the larger the radius, the more the water can move more quickly. And pi is just a mathematical term. The equation below is another equation that's important to understand, in which it considers the flux or the rate of flux is dependent upon two factors. One is the water potential gradient, which is, say, psi W1 minus psi W2, which is equal to the delta psi W or water potential gradient. But we need to consider the pathway. And there could be resistance in the pathway, and that's what the little r stands for. It says that the greater the resistance, the slower will be the rate of water movement. So the opposite of resistance is conductance. So it's the inverse of resistance, and that we use the term L. So we can use the water potential gradient divided by R, or it's equal to the water potential gradient times L. And this is important when we talk about things such as membranes. In terms of fluxes, we can say that something can have resistance, it cannot cross the membrane very easily, or there are pathways through that membrane, channels that will allow that substance to move through more readily. And therefore, we can talk about its conductance, how easily it moves across that membrane. So these equations allow us to calculate a flow rate or a flux. And the units here are in meters per second. The hydraulic conductance is a measure of the pathway permeability or its resistance, and measures the ease of which the water moves through a complex pathway, like through the xylem or a membrane. If we look through a membrane, then the L is substituted for an L subscript P, which is referred to as the hydraulic conductivity. So it's slightly different from hydraulic conductance, but it's not really relevant or important here to go into those very technical differences. Okay, so let's run through an example before we end here of water uptake, so that be sure that you understand the principles of water movement across a membrane from outside of a cell into a cell. And in this example, we see a cell inside of a beaker, and we can measure the water potential outside the cell in the liquid. And in this case, the water potential is 0 because the water is pure. So we have an open beaker here to have it at atmospheric pressure. And we'll just, for now, define that the water potential inside the cell is lower than that because it has components in it. It has solutes, it has potassium, it has sugars. And therefore, and by definition right now, we're going to just say that that water potential is at minus 0.2 megapascals. It's less pressure Remember, it's a negative pressure here. It's a negative water potential relative to the pure water. And so which way would water move? Well, we use our equations to understand that. And we find that the water potential of pure water is higher than the water potential inside the cell. So water would flow from the outside and into the cell spontaneously. So the rate of water transport, as we just defined, into a cell depends on the water potential difference, which helped us define the direction that water would flow spontaneously, but it doesn't tell us how fast it's moving. For that, we need to know what the hydraulic conductivity of the cell membrane is to determine the rate of transport. So in this example here, the initial differences were 0.2 megapascals. And I'm going to give you an arbitrary number of a hydraulic conductivity measurement, which is minus 10 to the 6 meters per second per megapascal. And if we use that equation that we saw in the example before, we would see that the flux is equal to LP times the water potential gradient. So we would have the numbers of 10 to the minus 6 meters per second per megapascal, the hydraulic conductivity term, times the water potential gradient, which is 0.2 megapascals. So we would have a flow rate of water of 0.2 times 10 to the minus 6 meters per second. Now finally, if we look at this last figure here, we can see that the rate of water movement will change with time as the water is being taken up into the cell. It decreases in time, leading to a slowing of the rate of water uptake. This effect here is that over time, this decline is decaying over a half time of that depends on the following parameters. Why is this important? Because later on we can use it to measure the hydraulic conductance or conductivity of a system and determine why water uptake in one plant might be faster than another might be due to its resistance or conductivity of the pathway. So this formula here, which we really won't deal with, but I give it here anyhow, just for those who are really nerdy and want to know how to figure these out. Perhaps you're a scientist. You can use this equation here that the half time is equal to 0.693 V, which is the volume, divided by the surface area, in this case of, say, a membrane, for example, times the hydraulic conductivity times, in parentheses, the volume elastic modulus minus the solute potential. Okay, I said I wasn't going to give you this term yet, but this is the last slide. We will define what that solute potential is in the next lecture.
Well, that's it. That's the end of lecture 2. In this lecture, we've covered the special properties of water, the polarity of water and its impact on its properties, as well as the formation of hydrogen bonds. In addition, we've discussed how water moves over short distances, such as diffusion, and also discussed the principles of the water potential, which we will use in the next lecture on long-distance transport. Don't forget that we will also have a quiz at the end of this lecture for the lecture 2. It'll be called Quiz 2, and there'll be a second quiz video with answers that you can check your answers and see how you did. Well, I hope you enjoyed this lecture. Have a great day.